Hello everyone, welcome back to my channel Xgene and proteinX. In this tutorial video, I'm going to talk about the overview of the cytoskeleton and also the protocol for using F-actin to visualize the cytoskeleton of the cells. Let's begin. So cytoskeleton is a structure that helps cells to maintain their safe and internal organization. It also provides mechanical support that enables to carry out functions like cell division and cell movement. Cytoskeleton of eukaryotic cells is made of filamentous proteins, and it consists of three major classes of elements. First one is the microtubules, and microtubules composed of protein, that is tubulin. And second one is the actin filament. These are the smallest one, and these are made of a protein called actin. And the third one is the intermediate filaments. These are composed of different subunit of proteins. And the tubulin contains two polypeptide subunits, and dimer of these subunits string together to make long strand called profilaments. 13 profilaments come together to form hollow straw-shaped filaments of microtubules. And these microtubules contain 13 different profilaments. Next, have a look the actin filaments. Actin actually is the abundant protein in all eukaryotic cells, and there are different kind of actin filaments network. First one is the cytoplasmic contractile bundles, microvilli, lamellipodia and philopodia, cell division contractile ring. So this different kind of actin filaments network having different kind of functions, like some lamellipodia and philopodia, these are for cell movement, and also for cell division there are some kind of it gives strength for the cell division. And also in case of the cytoplasmic contractile, that it contributes for the cell's contraction. And also, like in skeletal muscles, actin filaments slide along filaments of another protein called myosin. And this myosin it has two different structure, like myosin molecule and myosin head. And the function of this myosin is to make the cells contract. So next is the phalloidin. Phalloidin, it belongs to a class of toxins called phallotoxins, and which are found in the death cap mushroom. The major symptoms of phalloidin poisoning is acute hunger due to destruction of the liver cells, and mainly it binds and stabilizes filaments F-actin. Due to its selective binding to F-actin, it is widely used to visualize the F-actin in research. So phalloidin containing different kind of the fluorescence tags, different color of fluorescence tags, and because of that, by the fluorescence microscopy, we can visualize the different color of the cytoskeleton, like green color, red color, and blue color and violet color. This kind of different color are available for the F-actin. So these are required for different kind of the experiment. And next is the protocol for using F-actin, that actin staining agent. So first is the preparation of F-actin staining solution. So in case of this, 1.5 ml tube, you have to mix F-actin with blocking solution 1:500, and this you have to calculate that how many samples you have. The first, you have to take out the cells and wash the cells twice with PVS, and next fix the cells with 4% PFA, and next is the permeabilization, and then to wash the cells and then add F-actin solution that you prepared. And incubation time for F-actin is 45 to 1 hour at room temperature. And in this step, you have to cover your plate to protect from light. And then you have to wash the cells and then add DAPI. So in the same time, you can visualize the nucleus as well as the cytoskeleton of the cells. So most of the cases people do, like when they do the immunofluorescence, and in case of the secondary antibody incubation, in the same solution people add the effectin. So if you do fluorescence IF, then you can do that. You can mix with the secondary antibody solution with the effectin. But if you don't do the IF, so only the effectin stain, then you can follow this protocol. So after that, eventually you have to take that cover slip and use for the imaging. So normally people use the parafilm. So first actually, so if you culture the cells on the cover slip, so everything before effectin steps, everything you have to do the oils, like washing, fixing, everything you have to do on your plate. But just for the effectin, what you have to do, that you have to take parafilm, and then this parafilm need to be adhered on the table, and then by drop wise you have to place this effectin solution, and this drop, so one drop would be the for one cover slip. So you have to drop for according your number of the samples, and each drop contain only 40 to 45 microliter of the solution. So for this it needs very less amount of the effectin, and then the cover slip you have to put on the drop, and remember that the cell surface must contact with the solution. So by this step you can use only very less amount of the effectin, and this is very easy steps. So I hope this video will be helpful. If you like this video, kindly hit the like button, share it, and please subscribe my channel. And if you have any queries, kindly write in the comment section. Thanks.